It's better.
Is that better?
It's fine for us now.
Is that
Thanks.
okay? Okay, I'll try and do it this way. Just let me know if anything goes wrong. Okay, so to get started then, so I'm Dr. Emily Clark. I'm a research group leader at the Roslin Institute in Edinburgh in Scotland in the division of genome biology. So I'm going to talk today about the FAANG to Fork research study and some work that my group's doing at the same time. And talk more broadly about how highly annotated genomes are a resource to improve farm animal production. So just to give you an overview of the content of my talk, so I'm initially going to introduce why addressing the challenge of food production in the 20th century is a big issue at the moment. And then I'm going to talk about how highly annotated genomes can help to meet those challenges. Give an example, which is the FAANG project, and then talk about why understanding tissue-specific gene regulation is important. And then talk about some work that we're doing using allele-specific expression to empower sheep breeding, and then finally talk about emerging research priorities in the FAANG to Fork research strategy. So just to begin by addressing the challenge of food production in the 21st century. So the Food and Agriculture Organization reports that by the year 2050, the global human population is likely to reach 9.7 billion. So as a result, sustainable improvement, efficient production of farmed animals to provide healthy food and enough protein for this growing population are needed. So meat production globally is not showing any signs of slowing down, just what you might have, I guess, seen in the media. Therefore, we need to work on solutions that are sustainable and climate friendly to improve food production farmed animals. The challenge is to produce more food using fewer resources in a way that meets societal expectations around animal health and welfare. So highly annotated genomes from animals can help to meet these challenges. And improvements are required in animal breeding in order to do that. And chief among the improvements required is the ability to more accurately use animal's genotype to predict its phenotype. So advances in the analysis of genome function provide tools and knowledge for the genetic improvement of farmed animals, and the FAANG consortium, or the Functional Animal Genomes Consortium, is an international consortium working to discover basic functional knowledge about the genomes of farmed animals. And this knowledge will underpin genotype to phenotype questions in the farmed animal species. So the FAANG project is an international effort to characterize the functional elements of the genomes of farmed animals, and the first stages of FAANG focused on creating comprehensive genome annotation maps for a small number of reference animals for the main farmed animal species. There were 3 projects funded through the Horizon 2020 research framework, and they started in 2019, AquaFANG, BovReg and GeneSwitch. And since then, 3 new projects have joined under the EuroFANG umbrella, and these are Rumigen, Geronimo and Hollow Ruminant. So the aim of EuroFANG is to leverage functional genomic information to make animal production more robust and sustainable in Europe and beyond. And there are similar initiatives like these projects in the US, including the US Cattle FANG project. Oops, sorry. So in order to annotate farmed animal genomes, we need to generate functional information, and that takes 5 main forms. So the one that this workshop's focused on is obviously chromatin accessibility and ATAC-seq, and then also histone modifications in the form of chips to DNA methylation information about methylated regions of the genome, and then information about how the genome's expressed by characterizing the transcriptome. So FAANG projects have generated information like that. For example, the BovReg project that Krista mentioned. And I'm just going to give you an example of another FAANG project, which is the ovine FAANG project. This project's funded by USDA NIFA and led in the US by Brenda Murdoch at the University of Idaho. And the aim of the ovine FAANG project is to develop a deep and robust set of transcribed and regulatory elements in the sheep genome. There was a new genome for sheep released at the very start of this year, which is version 2 of the Rambly genome, and this genome is based on biosequencing, Oxford nanopore sequencing, and scaffolding using Hi-C. And the aim of the FAANG project was to provide a very high-resolution annotation of this genome reference assembly using the 5 types of functional information I described on the previous slide. With all of this data, the FAANG project will support annotating the Rambly assembly and make this data available to the livestock genomics community. So the new assembly was based on 2616, who is a Rambly ewe in the picture at the bottom of the screen. Oh. Sorry, that took a while to load. So the work that my group did within the FAANG project was to use CAGE sequencing to generate a map of transcription start sites across the genome. And I can see at the top of the bottom of these slides are cut off, but hopefully they're okay for everyone. Just let me know if they're not. The work that my group did in the FAANG project was to generate a map of transcription start sites across tissues, and these were tissues that were collected from the Rambly sheep, Ben's 2616. There was about 56 samples, and they're shown in the figure on the left. So we mapped transcription start sites using CAGE sequencing, which is cap analysis of gene expression, and we were able to map transcription start sites across tissues, look at those that were shared across tissues and those that were unique to specific tissue types. And the vast majority, as you can see from the figure on the left, were mapped to promoter regions. So variants within or near transcription start sites can change gene expression and can have a profound effect on the phenotype. For example, there's a mutation in the 3' UTR region of Texel sheep that causes the phenotype in some Texel animals. So mapping these transcription start sites is important, and we were able for the FAANG project to generate one of the highest resolution of transcription start sites to date, and we just recently did that for the Bovreg project using CAGE sequencing across tissues from 3 different populations of cattle as well. So highly annotated genomes are important tools to predict the effects of functional variants on complex traits and improve function. So just going to next talk about understanding tissue-specific gene regulation, and that did move forward. Functionally enriched genetic variants can be used to predict complex traits in farmed animals, and a detailed understanding of tissue-specific gene regulation is important because it allows us to tweak cellular processes, improve the accuracy of genomic selection, and also enhance efficiency, growth, and health in farmed animals. So this is a very simple diagram of genomic selection. So you have populations of animals with thousands of genetic differences and characteristics, and you can associate their genetic profile characteristics, so their genotype with their phenotype, and generate a prediction equation, gather genetic profiles, and then apply that prediction equation to breed the best animals. Functional information does allows us to add a layer of predictive power to the genomic prediction equation based on information about the how the genome is expressed and regulated. So we, for example, can include expressed variants or variants in regulatory region as weighted values for making breeding decisions. And our colleagues in Australia have really led the way in doing that. So this is a paper from 2019 where they quantified the contribution of sequence variants with regulatory and evolutionary significance to predict 30 different traits in Holstein Friesian cattle. So what they were able to provide was a framework to rank sequence variants based on their contribution to regulation and variation in different phenotypes. And the functional information they used included ChIP-seq information, methylation information, and gene expression data as well other sources, I think. So their sequence variant ranking showed consistent performances in genomic prediction of phenotypes. So it worked basically, and by providing public, so they used public data for functional information on genomic markers and demonstrated that their framework could make the global selection of animals more efficient and accurate. So my research group a few years ago generated a gene expression atlas for sheep, and we were hoping to use gene information in the same way that the Australian group did to harness the predictive power of functional information in genomic prediction. So we had this very large gene expression dataset for sheep. So we were able to create this atlas from a comprehensive set of tissues from multiple developmental stages, and basically created a searchable database of gene expression across tissues. So this is the atlas on the right. Each little ball is a gene, and each line is how connected it is to other genes. And the atlas highlighted key genes involved in development of the GI tract, innate immunity, and early development. And we used a similar approach to create a mini atlas for goats. So we had all of this information, and the next example I'm going to give you is how we wanted to harness that information to empower sheep breeding. So just to give you an indication of how important sheep are in the UK, there are actually 33 million sheep in the UK, and the sector is worth around 290 million. We're actually the third largest exporter of sheep meat in the world after Australia and New Zealand. So the industry is plays a big part in the UK economy. So again, I don't think at the top of this slide, but I keep going. The functionally enriched genetic variants, it's been shown by our Australian colleagues that functionally enriched genetic variants can be used to predict complex traits. The aim was to use gene expression data to predict muscling and growth traits in sheep. And we were using the tissues that were collected for the Sheep Atlas project to look at expression eQTL and allele-specific eQTL. And allele-specific expression is an imbalance of an expression in an allele from either parent. So the tissues that we collected for the Sheep Atlas were from a disparate cross, so the Tex has a double muscling phenotype, and the Scottish blackface that doesn't have that phenotype. So we expected an intermediate muscling phenotype in the F1. And this was some preliminary work we did for the grant, looking at the distribution of loci exhibiting allele-specific expression across the genome. So this is all 25 chromosomes. And each little dot is the ewe, the lamb, or the ram. And you can see imbalances of expression on chromosome 18, which is the well-known textile muscling QTL from the ram, and then larger imbalance of expression on chromosome 2. And what we have now is the power genome-wide these imbalances and how we can harness them for animal breeding, which we didn't do when obviously the textile muscling QTL was discovered, and we didn't have these sort of whole genome technologies. So, and our Australian colleagues again are kind of leading in this space really, and they've used an eQTL dataset from muscle and liver tissues in sheep, and the next stage of our work is to leverage our gene expression information with the information they've collected at scale. We just have a few animals, but they've managed to identify regions of the genome associated with growth at population scale level. So just to summarize then, just to finish, I'm going to talk about emerging research priorities before summarizing the presentation. So with colleagues in FAANG, including Krista and Elisabetta who are on this call, we put together a research strategy framework for FAANG research for the next 10 to 15 years, and we called it From FAANG to Fork. And this was sort of a set of 6 priorities. So the 6 priorities include high throughput in vitro systems, so having systems using gene editing to functionally validate, express and regulatory variants at scale in cell lines. And then developing pan-genomes and comparative genomics to discover, preserve and utilize genomic diversity, including, for example, the graph-based genomes that have been published by colleagues in FAANG. And then the third priority was FAANG GTEx projects that were very similar to the human GTEx project, and this has been taken by by colleagues in FAANG GTEx, characterizing genotype to expression information in large cohorts of animals. Technology is now available to collect large sets of phenotype data and then link that to functional information. And then the fifth and sixth research priorities were developing biobanks or in vitro biorepositories for organoids and cell lines that represent breed, species and population diversity, and then building single cell atlases that are that create life stage specific maps at single cell resolution. And together all these 6 research priorities should feed into gene editing, genomic selection, genome enabled management in a future where high value animals have their genome sequenced and management decisions based on that information, and then provide information about fundamental biology that links cell tissue and whole animal scale knowledge. And together all these could help breed healthier and more improved animals based on high quality annotation of farmed animal genomes. So just to summarize then, we've moved in the FAANG consortium, we've moved from small numbers of reference animals to associating large scale genotypes and phenotypes in animal populations. Sorry. To predict genotype to phenotype relationships. Public investment in international collaboration and training scientists is needed to expand genotype to phenotype research, particularly with industry. So for example, having exchanges with data scientists to spend time in industry and return to academia, and perhaps vice versa. So all of these things together will contribute to genetic improvement of diverse farmed animal populations to feed the global consumer, and allow us to harness the power of functional genomic variation and turn it into sustainable genetic gain to meet the challenges of food production in the future. So just to thank the people involved in the work, Sinead and Mazdak, who are in my group, and Mazdak's going to talk a bit later about our work in TACSEQ in pigs. And then the Ovine FAANG project led by Brenda Murdoch and is a collaboration with us in Australia and New Zealand. The FAANG to Fork concept was initially established with Elisabetta, Chris and Mick, and then Krista's the coordinator of BOVREG, as you heard. And then lastly, this is a link to the FAANG to Fork research framework if you would like to take a look at it. So thank you for listening. Sorry for the technical hitches.
Thank you, Emily. Don't know if there is any questions, maybe first from here, from people from Limoges? No? Okay. And I don't know if online in the chat box there is some questions. If not, people can just...
yeah, there is one question there.
Somebody...
How scientists without bioinformatics skills can access your data?
Did you see the last questions, Emily? So how
I can't
scientists...
see, yeah.
You can't see? I will read it again. So how scientists without bioinformatics skills can access your data?
Without bioinformatics skills. So the FAANG data portal's pretty interactive. You can just click on it and download data, and then to work with it. It would be difficult to do that without bioinformatics skills, I think. There are software packages like Galaxy that are point and click packages that you can input your data into, and then work with them. And then I think one of the nice things about FAANG is that there's opportunities to network with bioinformaticians through the FAANG task forces. So one thing I didn't mention is that there on the FAANG website there are new task forces now that are set up. So there's one on single cell, one on comparative genetics, one on farm GTEx, and if you go to join those task forces, you can perhaps meet scientists that can then help you work with the data if you don't have the bioinformatics skills yourself to work with it. If that's helpful.
I might also add that on purpose we are doing these NF core based pipelines because to some extent, at least with a limited amount of bioinformatics skills, people should be able to run rather standardized and reasonable data analysis from the raw. That's also one of the purposes why we put a lot of effort on these Nextflow and NF Core based data analysis. And people on the course will probably hear a little bit from Jose, but it's really a nice way. It's not click and drag, but it's at least still packages which come without the need to install all the different bioinformatic pipelines and softwares, and it's all packaged in a reasonable and reproducible way to make use of the raw data. So it's an intermediate. It's not something for complete newbies, but it's at least something even somebody who didn't study 5 years bioinformatics can make a lot of use with the data.
Yeah, that's great. Thanks, Krista. Good point.
And Mazdak just added in the chat box that you can browse, at least you can browse the cheap gene expression atlas. And he gave the link to check this. Okay?
Yeah, that's true.
And we can't see any other questions in the chat box. So thanks again, Emily. And sorry for the delay. Okay.